All right. Hello, everyone. Russ of Aquarimax here. And I have a guest, as you can see. Derrick, welcome.
Hello.
Thank you for coming on the show. I forgot to ask you in the tech check how you pronounce your last name.
Niehaus.
Niehaus.
Yep.
Niehaus. Okay. Very good. All right. Well, we have lots of people already here. We have Frank the Tank, Aunt Sandy Skinks, Mystic Drake, Invertebrate Person, Austin, Sean Meister. I'm probably missing some too, but there are people who are here. So young lads just joined in. Thank you all for coming. Derrick, as you may have seen if you saw the preview and the description, Derrick has done a lot with breeding Isopod Morphs, with understanding the genetics and so on. He's going to give us a lot of information on that topic today. So, Derrick, just to give us kind of some background, how did you get into that particular aspect of the hobby?
I think it probably started with Wally Kern's videos or something, just trying to show us isopods and how easy they were to keep, and just different type of YouTubes. I think it was right around pandemic, or it was a year before pandemic. I was looking for a new hobby, and I was like, "Ooh, this is going to be an easy, low cost, low maintenance hobby to do." And then it started with just, I think, one order online, and then I started collecting all around me. I was in Portland, Oregon at the time, so it had quite a variety of like Aniscus ocellus, Porcilius caber, a few Armadillium vulgare, but found like a yellow calico scaber and got really excited and tried to isolate that one. And then after discovering in my lottery mix how I had thrown in some pied into whatever the lottery mix I got, out popped an orange pied. And then I was like, wow, okay. And then I started talking to Kyle Calidian, or Chaldean. I don't know his name.
Candillian.
Candillian, thank you. He told me about just kind of how it's going to be a pain to try and isolate it out of the lottery mix because it has some unknown genes in it. But it turned out to be kind of a blessing in disguise, because then it popped out a yellow pied, and then it started teaching me more about what was made up of that, because I outcrossed it and got different ones out of it. Then I started making and combining my own isopods to try and get desired traits out of them. So that's where I guess the love for that kind of Mendelian journey to try and understand more genetics.
Cool. Yeah, it sounds like you've been up to some fun things, and I've seen photos. You sent me photos, I think, quite a while ago originally, of some of the things you've been working with, and then more recently we got back into contact, and I saw some of the things you've been doing, and yeah, you've got some really fun projects going. So I'm excited to see and talk about what we're doing now. So
thanks.
All right. So do you want me to pull up the presentation? We can kind of go through that, and we'll also field questions as we go and whatnot.
Yeah, sounds great.
Okay, here it comes.
Okay, so I wanted to give a little brief overview for isopod genetics, just in case anybody was unfamiliar with the different terms that we might use when talking about different types of morphs. Now, usually these traits would be in all animals, and we see them a lot maybe in snake genetics, different kind of mammals, but specifically in isopods, we're going to narrow down kind of what these traits would look like and represent. So we have our albinos, and some people get mixed up between the T-positive, T-negative albinos. The T-positives would be because they have tyrosine in them. And tyrosine is the precursor to melanin. It's kind of the starter pigment that'll be formed. And that's why we get kind of a reddish, maybe pinkish hue, but it always has some kind of light eye color to it. And that's where we get our tyrosine positive animals displayed. On the other end of it, if there's a mutation that happens on that tyrosine gene production, so it wouldn't even produce melanin because it doesn't have the tyrosine precursor, you get no pigments. And the T negatives will be our whiteouts, the completely white, no eye pigment ones. Those have popped up probably in a lot of different morphs now. I think most recently I saw some panda, some armadillidium, I think somebody tried to post spatulatus, but I think those were T positive. So it's good to see more and more of those recessive genes showing up.
Yeah.
And, usually it just may happen randomly through a certain isopod that has a mutation on that gene that will either cut off the melanin or cut off the tyrosine creation process, and then resulting in these different morphs that pop up. You also are able to find leucistic, or people call them Lucy morphs, but those are the ones where you might get like blue-eyed ball pythons or black-eyed animals as well, but they have every other part of them just has no pigment except maybe their eye pigment. We get those in kind of a few different morphs. Most of them might be mistaken for low expression Dalmatian. So you have your Lavis white, which are just dairy cows that have been line bred to have no other dots except their eye pigment. And sometimes some scaber morphs, which just may be scaber Dalmatian that has the low expression on them too. We have our orange morphs that come out. Usually you'll get some brown ones, or the browns will be the hets, and they'll pop out like an orange one, which I think is due to the carotenoids in that gene, or carotenoids are expressed, giving out that orange expression there. The melanistic gene is whenever you have like an increasing melanin gene, the overproduction of that melanin, and it just makes completely dark or black animal. We may have more representation in other species and animals, but maybe like the lava, that might be the closest one to just having a really, really dark one, like the really dark lava contenders. But I think lava is probably a mixture of a couple genes if you talk to some experts there. We have the axanthic missing yellow pigments. We have piebald, which is kind of any kind of a pattern there. I think the one pictured would be considered an extreme version of piebald, maybe even koi. So piebald, koi, Dalmatian, they're all generally all just a piebald gene, but they may be expressed in different ways. Like our Dalmatians have either a lot of little spots with the US Dalmatian and some big spots. The orange Dalmatians came from just orange, and they have just big spots, not the little spots there. And koi seems to be, you can't line breed or kind of produce a specific trait. It's just a random mash of all the genes there. Kyle's even tried crossing piebald with koi and just ended up with just more koi. So it's kind of where a random pattern morph where it can have low or high expressions of certain ones, but they just have pigment lost in a bunch of different places, and not really a set structure. As like Dalmatians, you can kind of tell with their smaller dots or spread out of dots. And piebalds, we'll talk about that later here. It has kind of some familiar traits that you can recognize. So I think we've already gone through here the tyrosine positive, tyrosine negative, different versions. Lava, it may be allelic. It might even be two different scaber subspecies that may have crossed. I think it's incanatus and then scaber that crossed, and that's where we get like a interbreed of those two, and that's what they proposed is lava.
Interesting.
And let's say calico. Here in the U.S., we have a sex-determinate calico where only the females will express it, kind of same with calico cats and such. I've seen a lot of red, orange, yellow calicos. And in the U.S., the yellow calico is probably the hardest ones to find. They are all in a mixture. If you get the orange calico, though, that one is a male and female calico morph. At least you can kind of line breed it so the males also have that calico gene expression there.
Right. Yeah. My understanding was that that's what Oren did, and that's why they're like that. When he originally got them from the Spanish stock, they were sex linked, but he kept breeding them until it showed up in the males.
Nice. Yep. That's good backstory. I didn't know. But that is definitely one of those where it has that sex determinant dimorphism, with our US ones, there are also some different lines that also have that. The way that you would try and get a recessive trait out of something is, let's say we're going to like try and cross them, or how is that recessive trait expressed? Well, with Mendelian genetics, you will, so you have a small r, big R, big R. Whenever you would mix them, everything is going to be big R, small r. So they're all going to be hets, and they won't really express the recessive trait any longer. For example, whenever I made a U.S. orange pied, I used some orange males, and I used some female Dalmatians, just so I can mix them together. You would then get those double hets, and you want to try and isolate just those when they're juveniles, so they don't try and cross with their parent genetics, or they want to just cross into themselves, kind of to interbreed. Those babies then have a 25% chance of ending up with that small r, the recessive trait that you're looking for again. And with two different kind of visuals, you will get that visual trait, pull them when they're young before they have had time to cross with other double hets. That way you can prove out that line. You want to then inbreed that one again, and you'll solidify or kind of prove out that trait.
Yeah, that's a great explanation of it. I've done it several times myself, but others, I think a lot of people have never done it and need that step by step to be able to do it. So that's awesome. It was very concise way of explaining it too.
Yep. The problem that some people might see is if they leave that in there alone, it might start to cross with other ones, or they've tried to get more of a trait. They found one visual, and now they have as normals left over. So they would put it in the mix, and then they'd have the babies in the mix. They'd leave them in there, and then they might backbreed with the adults, not really getting that visual trait that you're wanting to pop out again. You might get very small amounts. So if you would just isolate the next generation and then isolate the visuals again, you're going to more quickly prove out your traits. You won't have sometimes either hidden hets there that you can't really tell because they'd look just normal or wild type still. We have some different types of double recessives. These would be kind of ones that exist and some that I've made. Orange Dalmatian is out there as kind of a good one. I think you made a nice orange Dalmatian cross, and I'm trying to do my own. It's very more expressive. It's got the small little orange dots, whereas the other orange Dalmatian line that's out there that came just from the orange line doesn't really have those. Orange Pied is the one that I've gotten out of the lottery mix, isolated, and I've also created one from US stock. I've also recently made some lava Dalmatians. Those are fun. It's a little bit difficult to tell what's just regular Dalmatian and what's a lava Dalmatian unless it has that orange patterning on it as well. But
in the larger spots, yeah.
Yes, those look pretty good. And sometimes you'll get really high expression Dalmatians on them. So that's what I'm trying to get more of that line bred to get the higher expressions before I try and release them. Lava Pied has been kind of a harder gene to isolate. I don't know if it just my colony is not producing as well as the lava Dalmatians are, but I've gotten a few visuals that I've gotten to isolate now, and they're starting to reproduce.
Oh, good.
Mm-hmm. You can have Pied Dalmatian. I've found a couple of them. It's kind of hard to tell where you just get a few spots on a few segments. Mm-hmm. I have a picture here later where I'll show you where I've made a triple recessive with them. Yellow, that is I think it would be as similar to yellow snow that's out there, but I can't say for sure. I've not outcrossed it to tell if that is what it's made up of, but I have had other indications that yellow is a mixture of T-negative albino and orange, where you get a yellow pigmentation.
That's cool.
I've also mixed the T-negatives and T-positive albino vulgari, trying to get like a more peachy color there. It just kind of looks similar, just like a higher expression or like a lower expression T positive vulgari, but just different kind of doubles that you'd have to do that same process of crossing them, isolating the normals, double recessives, and then intercross them again. And to kind of create those double recessives, as I was talking about it there, we'll do the same kind of thing, pull them aside, and then inbreed or just keep breeding those selected ones to solidify that trait. And this photo is kind of one of when I was trying to isolate the orange pied. It had already bred with some other unknown gene, but it created this big multitude. I think that's ball try.
Oh, okay.
Those are calico in there. They've got the regular pied. And I don't know if they pop out any orange pied. I don't know if that gene's in there is more of it's just a calico pied, but they kind of express that line.
An interesting point that you just brought up, we need to make sure that everybody catches that.
Sure.
Is that when you are breeding isopods for specific traits, not only do you have to isolate the young, the next generation, like isolate the F1s and breed the F1s to the F1s, when you do it, you have to select them when they're quite small because they can mate when they're far smaller than adult size.
Very true. That is a key there. You don't want them to try and cross with another version, double head or anything, because then, you don't want to have them to backbreed or cross with another double head because then they won't have that pure visual offspring. And it's going to be harder to isolate that line out. And you might end up with little tiny babies that are still in there that don't have your visual trait. So one thing I've done with the orange Mardi Gras pictured here on the left is I've always pulled them. They're very easy to see the visuals when they're so young and pull them and then isolate them into a separate bin. So that one should be really clean to try and isolate out. The bottom middle is kind of some lava pied that I had. I'm unsure if the other ones are just regular piebald, but I've tried to just start to isolate just the orange and black pied. You can tell that it's lava and not calico because usually it'll have that black tail. All the lava usually seems to have kind of that black middle, has a black dot on it, or just a full black tail for it. Mm-hmm. And the top and the right photos are from my experiments with U.S. orange and U.S. piebald. Instead of my last ones was using a Spanish orange, and it always had this. In the top right, there's a male there that has this really bright or really clear white middle. So kind of like an Oreo, where orange head, orange tail. But if you also notice on the face, it's got a little bit of missing pigment on the cheek there, which is different than the other pied line that I have. The females seem to be coming out a little bit more coy looking, where they have more randomness where their pigment loss is. So I'm excited to see how this line differs from the other one that I'm breeding out pretty consistently, where they all have that middle stripe that is missing that pigment. And I think those are a really sharp looking line. It kind of looks, I don't know, the jaggedy edge, trilobite-ish. I just am drawn to that. So hopefully I've given it to 3 other people, Kyle, Jessica, and I think it's Ed. But we're hopefully going to start sending out more so people can have this. When I have some more, I can try to send you some rust too.
Oh, yeah. I would love that. That'd be awesome. I love the idea. And your photos with the that clean middle, like the kind of the Panda King, red Panda King version of Porcelio scaver. It's really cool looking.
Yes, that would be the closest visual thing that we have out there for it. Now, there is Jeremiah that already had made what he calls the orange Mardi Gras on the far left. I did the actual cross myself, where I took the maples and I took Mardi Gras, or there was kind of this different line that Kyle had isolated called Stardust.
Yeah.
And I crossed those, and I got my all brown ones, interbred those, and it took a while. I got some other Mardi Gras looking Dalmatians to pop up, and then boom, I got at least a couple of these visuals start showing up, and every so often one will pop back out and I can put it in this bin to isolate it. I'm hoping that mine it has that more yellowish expression on the back that maples don't really have. And it's got more of an orange expression than ones that Jeremiah has. I think what happened with his is he had a colony of maples, and similar to our orange Dalmatian scapers, they popped up, and he's isolated those Dalmatians out of just the maple line. So I don't think he actually did a cross with Mardi Gras and maples to get his. So I'm hoping that mine has that more yellow expression and the more coverage there on kind of the skirts. Kind of moves more than that.
Cool. Yeah. Then anything you can do to get that higher expression, that's awesome.
Mm-hmm. And some other things that what I was talking about earlier is those yellow morphs, where I have done experiments to realize that I mix in the whiteout gene or the T negatives with an orange gene. Mm-hmm. You get left over those carotenoids, kind of reduced tyrosine carotenoid. Instead of a pinkish color, you can get that yellow pigment. And in there is also a pied, so yellow pied, where it's missing that band in the center, triple recessive. I've set up an experiment where in the top middle, I've just done regular orange vulgari and T negative vulgari albinos, and working on those kind of offspring. I've also put in the orange Dalmatian, so like the orange magic potions with the T negatives, and I'm starting to get some that start to look yellow, but none of them that have yellow spots yet. So that'll be a fun project, hopefully getting 2 lines out of it. I don't have this one on the bottom right, but I saw somebody had posted it, this really highlighter yellow vulgari picture. Mm-hmm. And that is kind of my goal, to get the highlighter yellow isopods. I've made that cross in a bunch of different species now. I actually, if we have time, I'll show you a bin I have. I'm going to call them Sunshine. They are Aniscus ocellus maples mixed with the whiteout line, or maybe it has a little eye pigment, so maybe leucistic. But I'm trying to get just all yellow Aniscus ocellus. Ooh. I was able to get Florida Orangia cubaris marina, and I've mixed those with the whiteout glaciers. Mm-hmm. I haven't had any success in making a yellow yet, but they have what I think popped up their visuals. So I was wanting to call them yellow submarina.
I love that.
That would be a fun species to have, fun name. I think, though, the double recessive is kind of a loose whiteout form that's got a little bit kind of a purplish down its back. You can have more whitish skirts. I'm not sure that's proving out in the cubaris genus. So I've done the vulgari, scaber. Oh, I'm doing a pruinosis, Porcellinoides pruinosis, orange and whiteout. I've mixed those. I have the wild type ones. They're growing up, and once they start producing, I'll try and isolate out a more yellow one. The T negatives kind of look yellow as they are, but I'm hopefully getting more of that highlighter yellow whenever I get those out. I know it works well in the Porcellio lines. I've also inspired people in the UK or EU to mix their orange Hoffmannseggi with the whiteouts, which I think the US only has the whiteouts, and we're due to get the orange sometime, maybe. Mm-hmm. Hopefully they can come out with a really yellow Hoffmannseggi, and that will be fun. I won't be the first one to that, but any other genes that are out there would be fun to create. I've seen some different kind of genes out there that would just be nice to try and maybe the first one to get that yellow out of them. So some triple recessives. As I was talking in the last photo, you can tell that it's got that T negative gene in the top photo. That is my yellow pied. They were an accident created with the orange pied line that had a hidden T negative recessive trait in there, the hidden het, and every so often it'd pop out this white and yellow one. Has no eye pigments, so that's why I proposed it was the T negative. They have crossed out, and I've gotten just orange ones and just pied ones and just all white ones when trying to isolate them.
Wow.
The other triple recessive I've gotten from that same mix I think a Dalmatian snuck its way in there somehow, and I have tried to re-isolate the orange pied line, but I started popping up with these Dalmatian pied, orange Dalmatian piebalds. So if you can tell in the photo, they've got just specific lines where there's no pigment, kind of more in the middle, and then towards its front and back, they have those kind of Dalmatian speckling on there.
Oh, especially in the bottom left individual in that photo. Yeah.
Yep.
Totally see what you mean. That's
Just
cool.
one band. So those were an accident, but I'm isolating those as well. So even if it's not a pure cross to the two of them and you find a trait in your lotteries, you can try to isolate it out and get a lot more fun stuff. But these are the 2 triple recessives that I've created so far, more or less by accident.
Very cool.
I'm working on that triple recessive yellow dots vulgari, or yellow magic potions there. Yeah. One cross that I'm happy to have made, I didn't mention earlier, but I've created an orange Dalmatian lavis. As you were saying, the California mix, that might be some kind of a pigment loss through a non-lethal pathogen that causes those kind of randomizations. I tried to mix them with dairy cows. It didn't work. I tried to mix them with milkbacks, which you've done those crosses, and you know the results. You're not able to get them to make any wild type double recessives. I've heard somebody has done that in the UK, or the EU, but I haven't seen any results from theirs. These are more of a lower expression. The milkbacks, isolated by Kyle, they came from the US caramel line.
Oh yeah, the how nows. Yeah.
Yep. Sorry. Yep. That how now line mixed with our orange gave me these really brown lavis, and I knew I had something going on. They were able to get that genetic cross. I actually just took a couple, like 2 of each, 2 males, and then I think 3 females of another, and put them in a small 32-ounce deli cup, and then boom, lots of little brown double recessives came out. Mm-hmm. Then those inbred them and got these visuals. They have kind of an orangish glow coming from the inside, and then just some dots on the outer skirts. I've given them to some other people, and they've got a higher expression. Kyle now has them in his hands, and he's working on a higher expression of them. And a couple other people, they've grown out a whole bin of them. It's hard to try and delineate if it's orange or if it's just more how nows when they're really small, like the one behind it is a how now. Mm-hmm. And it's kind of, it doesn't have that orange glow coming up from the center. So I like that trait. I just hope we could get some more higher expression so it actually looks like some big orange spots instead of just a little hint of orange on the tail and sides.
Yeah, yeah. But it's still a very cool look. And these are the ones you call
Colby Jack.
Colby Jack, yeah.
Jack cheese.
Yeah, I love that. It's a really awesome name for them.
Yep.
So the U.S., the lavis that is in the hobby in the U.S., as far as we know, is true lavis and will cross with these how nows. Is that the stock you use, the orange hobby stock?
Yes. I think both of them probably came from the caramel line, is my understanding. I know the how nows are, but I don't have precise knowledge of the orange ones are. But they were able to cross, so they got to be something along that same species. Whereas now we might think that dairy cows and their milkbacks came from them. Those are probably another subspecies or a related kind of lavis morph species, but the parts don't mix. So that's probably the main reason why we can't get those to easily cross.
Right. And I have seen stock in Europe from orange lavis that just appeared spontaneously in orange lavis tubs, where it is a true piebald orange lavis with very stark white, big patches.
Nice.
It's very strong contrast. But I was trying to see if we could get some over here, but I lost contact with the originator. But the photos are awesome.
I've seen photos a while back. I've not seen anybody post them recently, but they had what looked like dairy cows in scaper, those kind of big black spots.
Oh, yeah.
Orange version of them as well. I was like, oh, that would be a great Dalmatian morph to get hold of.
Yes, it would. Like with the high expression of dairy cows, I've seen it. The Tunisian pied is one of the names they give it.
Okay.
But I have not seen them in the U.S., and it's kind of hard to track them down in even Europe these days.
Yep.
Because I would love to get those over here.
I'm waiting to see a Convexus Cyclicticus convexus.
Oh, yeah.
The Ukrainian pied. I'm waiting for them to get an orange one so I can make that cross, because they are really striking with their black and white patterning and get variation expression. So those are going to be a fun project if we ever find some orange ones as well.
Right. I hope we do. Or even just get some of the Ukraine pied, we get an orange mutation in there, and then the work would be done for us mostly.
That would be great. Another way that you can get those traits out of things is line breeding it. So it's more like increasing expression of certain things, getting that more patterns, or just these were some wild stock that I found in Oregon of some high yellow, and several, I put like 3 of them in there that had that yellow tail spike. It must have been happening in that colony that I'd come across where they get that yellow tail spike. I've seen it in Hasi, some other breeds that just have kind of that yellow tail, and it seems to not be consistent, but it will still more likely happen with the offspring. And I've been able to still get 1 or 2 pop up now that have that yellow tail, and they're starting to get really high yellow patterning. You want to always be culling out the low expression ones out of the bin, or isolating. After a while, I just pull the highest expression, put them in a new bin, let those interbreed, like the ones below it. And that's basically it. You just keep repeating the cycle until they get more and more condensed in that patterning. But if you let them go and you just keep all the regular ones in there, they'll start to lose that patterning and turn back to normal looking colors. So it's kind of a consistent project to keep working on, and probably reminds me I need to do another culling of it. They've gone down probably like 6 generations or so, and they're starting to look pretty nice. Like all the babies will have that higher yellow expressioning to them.
Cool. It seems like, yeah, consistency is key, and that is something that, a lot of people have interest in that, but in trying to isolate morphs. But then when it comes to the consistent work you have to do, that's where it really comes down to it. Can you maintain that effort and continue the effort? Because it does take generations, and especially if you miss something. Like I isolated a pied Porcelionides pruinosus from crossing oranges with Oreo crumbles.
Oh yeah.
But I still occasionally, because I'm not as consistent as I should be, I still occasionally find an Oreo crumble visual in there. It's mostly they're orange pied. It worked out, but occasionally I'll find one in there, and I really need to do is just pull out a bunch of really young orange pied individuals and get a pure culture going.
I've done that too, where they got in and they started reproducing more of those double trait, or those 2 different kind of ones, and I called mine a Halloween mix because it was just black and white. So that's where I left those, and I had another line. Actually, those orange cream ones, Kyle had created them. He had sent me those along with the Witch's Brew ornatices. I suggested the name Witch's Brew, so he's like, okay, I'm going to send you some. So I got some of those first ones, and me and him helped distribute those along with the orange cream pruinosis. He sent me, I think, a couple visuals and just hets, and then I proved out my own line, and he had his own line. I'm trying to more concentrate it so it just gets higher and higher expression of orange in mine. The one things that I've noticed is people kind of post images of things, and it's like, is this pied or is this white on them genetic? And so most of the time there's a way to pretty much tell if it is going to be a piebald, at least for Porcelio species and some others. When they're missing that middle bands of color, or like a full line or stripe of color, that one's going to be more pied. It needs to have at least what I've found, pigment on the very front head where the eyes are located, and then mostly kind of on the tail. That needs to still have pigment for it to be more of a genetic pied, whereas some other ones might be damage from whenever they were born. In the bottom left, I had some high yellow ornatus that popped out, these all white or leucistic ones, and then that one is a male that I've gotten, and it had kind of a little bit of patterning. It must have just been all from one clutch, and they might have had less nutrients as they were growing up, or something occurred while they were molting. But I tried to inbreed just those. I'd got them when they were really small, and they grew up, and I just got normals out of them, like maybe just more reduced yellow, like very, very low yellow amounts on that. But it was not genetic. Similar on the bottom right, somebody had posted a photo I grabbed that their tail is missing that color pigment, or it's kind of like not consistency. I don't think those are genetic. Those are more damages or maybe a non-lethal pathogen that's causing that.
Mm-hmm.
Similar in the top right is the Montenegro or Klugai Dubrovnik lines, where they kind of just produce that all white patterning. I've tried to inbreed those, I think, twice, but they wouldn't be able to produce. I think they're infertile, where they won't be able to pass along those traits. So sometimes they can create those that pop up, but I just haven't been able to consistently get more of them once trying to inbreed them, or they revert back to normal. I think the Dubrovnik line just came out normals after that. So there's something strange about some of those genes. But in the top right is my lava pied, one of them that I'd isolated. So I know that was from that morph or that double recessive cross, and I was able to get those. And I think Kyle has a better line than I, but I just wanted to make sure I did it and get those.
That's a cool effect there.
Mm-hmm. So more just when you're getting piebalds, you don't want to have inconsistent pigment loss in different places. It needs to kind of display those traits and have the head and tail plates have those colors still. I think the top right, where what ended up coming out of my yellow or yellow calicos, they kind of muddied up a little bit for those. But those are some different things I've been working on. The lava pied, lava Dalmatian are maybe some more recent ones that I've been able to prove out. And then let's see, I've got some of those bins here. But lava Dalmatian, let's see if I can show, just pick up the bark real quick and see.
Mm-hmm. Well, let me make this. I'll put this in solo layout so we can really see it. Okay, here we go.
So there's one down there that's got really high patterning.
Oh yeah.
Sometimes it's hard unless they have the orange on them. It's hard to tell that they would be lava pied. Can't really see. There's a couple. That one's got some orange. It's hard to tell in the video. Everything's a similar pigment.
Yeah.
So it's really good. I did another cross with orange Dalmatian and lava. So a lot of them are just kind of like an orange Dalmatian. I wanted to see if I could get anything looking different. They're not as cool as just the regular pied though. And here's the bin of the lemon pied. They've been sometimes popping out just all white out looking ones. So I always got to try and cull and get just the ones that I think have a little bit more yellow on them.
Mm-hmm.
It's maybe hard to tell, but they can sometimes come out all yellow, or you just barely faintly see a little bit of yellow on the heads and the tails. Like this one right here is more of like an all yellow or a very low expression pied. Maybe this one too. They're all just color of wood. But yeah, those are fun trying to line breed them.
That's very cool. I like that.
Some that I've come up with is called Lottery Ghost, that I'm giving it the name. But it is all the T negatives, and they'll be het orange, which will come out and be purple. So purple, yellows, some really kind of light orange, and even some pied. So I think this one on the edge is the pied yellows.
Oh yeah, you can see that pretty well on the screen.
Yep. And then this one in here is kind of a purplish.
Yeah.
Going that well, but that's the T negative gene working in there.
Mm-hmm.
I think that one is the more yellow pied or got some orange trait to him. I think that's going to be a pretty funky mix. I like them better as like a ghost. So this one is a het orange. So it's kind of like that purple head and tail, but it's also got pied in there.
Yeah.
So kind of a mixture of different colors in there with those. But that's how I knew that the T negative line was involved in making the yellow gene.
Cool.
Here is the bin or the original colony I have, the orange pied. Now, this is the one that's sometimes popping out, the orange pied Dalmatians. So if I see any that are white in there, I'll try and smush them or isolate them. But they have given me some just solid oranges when they're younger. So I'm wondering if that's just, I need to do another culling and re-isolation of them. But mostly they give out that orange pied. When they get older, it turns kind of more of a darker brown orange to them. And they lose more of the pigment in the middle. They can even become like all white on the top. And they're not as, so this might be an example. This one here, they're not as consistent as you would think. So I think this is an older one where they kind of get a little bit of orange pigments on their sides, or the white kind of spreads out more.
Yeah. Like
this
that
one here.
Yeah.
So it's kind of like the white pie starts to take over more of them. And this would be maybe a younger one where it has just those two little stripes in the middle, but they all have the pigment on their head and tails to give them that orange look or that pied look to them. So I know that's more of a genetic pied.
So what are the origins of the two different piebald lines that you're working with?
So this one came from a lottery mix, and it had a Spanish orange in them, and just I think a U.S. pied or a similar kind of a pied morph. So this one has gotten the more consistent orange lines to it. The other line of orange pied I've made is where I mix the U.S. Oranges from Kyle and the U.S. pied, and they kind of give me, the males give me that same kind of very strong pied middle, but the females are kind of looking like a more coy look to them. So I'm starting to get little bitty babies from those, let's say. This is the other one that I'm just starting to get double hets. I'll try and see if I can get them on visual, but it's the one I'm going to call Sunshine.
Oh, yeah. I remember you talking about that one, yeah.
So the other ones, I wanted to give them the name Maple Blossoms because they are white and orange, and then they have little yellow bits on them. So if you look at a maple blossom, it's like the bottom is an orange. They have white, like stema, and then the or stomatella, and then the tips of them got that yellow pollen look to them. So I wanted to get that name, but I wasn't the first to create the look. So I think they're just called Orange Mardi Gras now with Jeremiah's line. But let's see if I can find visuals in here when I lift this up. So this is using the T negative or the whiteout oniscus and the maples. And there are some tiny little like double recessive brown ones in there. I don't know if we can see. Oh yeah, there's, I think this little one right here is a more wild type looking one.
Oh yeah.
That size now versus this would just be a maple. This one is, so those two are going to be the ones that I'll try to isolate just when they're a little bit bigger. Try not to really, really tiny baby. But I think more maples were in this one, or I tried to get one sex of the whiteouts. There's a few visuals here, so I'll lift up and show. I think it's more of a leucistic because I see some eye pigment, but I don't know if this one will try to make that cross with the maples and more yellow morph out of it. So
it's really interesting that the whiteout reacts with the orange and allows some of it to come through. And that seems to be true of multiple species too.
Yeah. And I think that's where it might be just a different type of pigment gene, the carotenoid gene.
Yeah. Or maybe it's even sequestering carotenoids from its diet.
Mm-hmm.
Yeah. Who knows?
The maples and then this whiter form one. So I've got some double negatives. I was worried that the whiteout had just come from maples, so I was just going to get more maples. But I did get some wild types in there, so we'll see what the double recessive morph of that looks like, and if it does come out any yellower than those.
That would be cool. Highlighter yellow aniscus would be pretty cool.
Mm-hmm. Hopefully I can get that Vulgari line too. They're the strongest yellow that I've seen, at least from
those photos.
Somebody's gym mix. So I was like, oh, I know what that is. It was a gym mix and it popped up the yellow one. So I'm trying to mostly recreate that. I think I have a few visuals. They're not as very stark yellow, but it's definitely not the whiteout form, and it's a lot yellower. I need to isolate those. I think I've left them going for too long. But if anybody had questions of what do I feed, I just use tropical fish flakes, and then I make a little powder concoction here. So what is in this? It's green. I used to use spirulina, but now I use chlorella, and that's because it's a freshwater algae, and they'd be more freshwater isopods versus spirulina is a saltwater one. So I've had some different mix. I'm trying this one to see if it does any better. The reason why I'm adding that is because the blood of isopods is actually based on a copper ion instead of like hemoglobin that we have. So they would need more of that copper, and it kind of turns a greenish color when oxidized. But I will mix in. So this is the new stuff, just broken cell wall. That's probably what they're eating out of the leaves and other things sometimes. But in the center of every chlorophyll is a copper ion. And then I got some of the isopod chow, just some new, powdered mix there from Wally. So that's the base of things. Mm-hmm. And I added a mixture of calcium carbonate powder and just some animal vitamins. So then they can get all the different vitamins that they need. The calcium carbonate for their shells, they get that from hardwood naturally. So calcium is in hardwood trees. That's why we feed them a lot of that kind of wood. And so the powdered food mix, Wally's got them rice bran, cornmeal, flour, cane molasses, dried brewer's yeast, flaxseed, bean flour, green pea flour, cinnamon. This actually has some spirulina in it too. But I think that kind of is just a good roundabout nutrition for them all the way through. I've tried doing separately, like some bee pollen works well for them, and trying to think of other things I've fed them before. Oh, dehydrated carrots and just some dried out peas. They're organic, and I throw them in there. Any wet food seems to mold or mildew within a day if it's fresh. So if I am getting fresh carrots, I'll let them dry out after I've cut them up for a day and then throw them in. They seem to last a lot longer without mildewing, and they'll rehydrate or replump up. They'll still eat them the same if they're that freeze dried or those kind of veggies is what I more feed. So it lasts longer. And some people put like a pile there, but I think it was Kyle or somebody said if it's just located right there, how are the little baby ones going to get to it? If they can't climb over the edge of that shell or go to that specific location to find it, it's harder for them to find it. So I just sprinkle it everywhere, broadcast it all over so it hits the soil or the tops of leaves, and they kind of will be able to find it wherever the babies are in the tank and get their extra nutrition or supplemental food. And it's the best. You'll put it on there, and so like the top of this bin has still got some food, but it maybe has a little bit of green left in places. But when it's fully cleaned off, you know it's time to feed some more. That usually is maybe once every 2 weeks. It's good to have either cross ventilation, top ventilation, just something that'll allow that air exchange. I've got a fan going in there, so it can help to put some more fan movement. If you got the giants, they definitely need more ventilation than just smaller ones. Some can have very little ventilation in there, but they'll still be fine. I think more like pruinosis, some don't need as much vulgari, don't need as high of ventilation. Aniscus ocellus like a little bit damper. So it depends on what species it is, how many ventilation holes I'll put on them sometimes, or just whatever bin is left over after the last experiment, so I don't have to make any more.
Yeah, that makes sense. And I've noticed that a lot of different types of ventilation work for a lot of different isopods. Many times there's a lot of wiggle room there.
Yeah, I think so. And The best advice is cover the ventilation holes. If it's like a mesh, that'll let mites in, or little babies will try to crawl out. So the finest mesh material, or I just got a bolt of chiffon fabric and have never run out of it. Or you can go to your local fabric stores and get a yard of it for under $1 usually. Just find some, and then you got it out and you can make 30, 40 bins of it. So that's what I'd recommend, just putting over if you got any screens or something that just has large holes for ventilation. You want to make sure you can keep out the pests and keep in the springtails.
Yeah. Yeah.
I'll have these bins, and even with them like this, I'll find a bunch of little dead springtails or little tiny dead isopods. Like the Monkey will somehow find their way out. I think it's crawling up and over, so they don't have gasket bins. These are a little bit more wiggle room. As long as bins are on top, that can kind of help keep them down. But
right,
you can get some invaders that way if you just have your top of your bin and you flip it over, and all those maybe dead things drop in there, and you'll get your invaders. Most likely, like Pruvinosis, will find their way into bins. You're like, how did you get in here? And you don't want to start taking over everything.
Yeah, they're amazing for that. That's for sure.
Yes. Several bins have done that. Another project I don't really have visualized, I'm trying to prove out, is rubber duckies. I bought somebody's colony or line that produced kind of a white middle instead of it being black. So it's a little bit yellow on the fronts and ends and white in the middle. I'm going to try lemon drops or something else, or lemon candy. And what I found in there too, what popped out as well, is like a real pink ducky. They have yellow on the front and back, and it's a bright, or kind of like that Sakura blossom or the cherry marinas that are coming out. It has that kind of middle. When they grow up, it kind of more darkens to brownish, but you can still has kind of a pink tint. Whereas what is called the pink duckies now are just kind of like a muddled ducky, where they can be a little bit inconsistent pattern on the back middle or brown in the middle. But these have come out of the same one. Right now I'm keeping them together. So maybe if it's like a male and a female thing, sexually dimorphic on that trait, and we're trying to get 2 lines out of them, like the leucistic form, more yellow form of rubber ducky, and then that pink middle. So that might be pink lemonade.
Oh, I like it. I think I remember Kyle talking about a line, and he talking about you in reference to that line. Yeah.
I gave him some of those too. So he's trying to prove it out as well. He gave me a really neat sexual dimorphic line where it was the Cubaris red edge, but it had some T positive albinos and some orange ones, and they're sexually dimorphic. Like they're one and the other. I took some of those orange ones and I've mixed them with red edge whiteouts that I got from somebody else. Had them. They were loose. They popped out of the red edge. They were leucistic. They had the black eyes, and then those interbred. So I think that was the kind of hidden het with the leucistics, and they popped out just full T negative, colorless, no eyes. And I've mixed those 2 together, and I'm trying to get the yellow form of those too.
Oh, so that would be cool.
We'll see if we can get some red edge yellows.
Yeah, absolutely. Very cool.
So any yellow cross that I can try to think of, I want to try and make.
Yeah. I'm just kind of blown away by the yellows you've been talking about, and the photos you showed of those Fulgari that had just like you said, like someone took a highlighter to them. That's awesome.
Yeah, we'll see if we can get them in the hobby. It takes a little bit of time to get everything proven out and enough to then distribute to people, and then they have to grow enough to distribute. So as long as I get it into the right hands, they'll be able to start moving them. So I'm thinking Kyle's got them already. If you can get some of these morphs, like you send some to Wally, and I'm just trying to send the Smug Bug, Laura, next. And so then she has a very prominent website. She can start to display them.
Yeah.
And carry those too. So they're getting to that stage. You saw there was enough orange pied in there to try and send them out. The problem is once you pull a good number, it can sometimes crash out the colony. That has happened to the Maple Dalmatian or the Maple Mardi Gras.
Mm-hmm.
Those the original colony, I'd sent out some of them, and now it just kind of crashed out. And I've got maybe 3 or 4 in there. They're double hets and maybe maple, so they're going to start to rebuild. But luckily, I have the visuals. They're isolated, and they're going to start to inbreed and start to produce some. I've noticed very tiny babies, or they've only just started producing the next generation.
Oh, good. So,
maybe by fall or something, there'll be enough to send out to somebody, or I'll try to hold them back till next year so I don't crash that colony if I send out the younger generation,
mm-hmm.
Or send just a gravid female to Kyle probably first, so then he can start to work on. He's kind of a backup in genetics. I'll have backups of these genes, kind of like the U.S. orange scaber.
Mm-hmm.
He lost his colony, so I sent him one of the U.S. orange males and females that I still had left over from that project of making U.S. orange pied, and then he's trying to isolate it back into just the U.S. orange scaper.
Cool. Yeah, because I guess Ryan, the one who isolated the U.S. orange originally, is not really doing it anymore.
Yeah, I don't know where he gets some of these things, but I know that he has them. I know he didn't find the original magic potion, but he helped to push it out there and be one of the major distributors of it.
Right, because he refined it once. It was Jay Fiore who found it, and then he sent stock to Kyle, and Kyle worked on refining it. Yeah.
Yep. I think he did isolate the Dalmatians, though. Got him from Briceville.
Yeah, I think it was Alan John who sent him a stock or something. Yeah,
that's probably it. Yep.
And then he worked on that. He's done a lot of this. A lot of the isopods we have in the hobby come from stuff that he worked with. The whiteout P. pruinosis. I think he was the one who found the first stock there. A lot of them. I can't even think of all of them.
He's helped inspire me and been my major bounce back and forth to help me learn the genetics, and told me that shouldn't even try with the orange pied because it would be so hard. They got so many hidden hets in there and stuff, but I was lucky enough to just have that one hidden whiteout gene that just kind of popped out the lemons.
Mm-hmm.
So that was a nice find. And since then, I've done purposeful crosses here so that they don't have the hidden genetics and I can just get that trait. But hopefully I can become more prominent with my morphs out there like he has in the past. I think he did most of it. It was 2011 range or something when he was starting to create most of those things.
Yeah.
And I just want to make my name and put a few morphs out there. And if I can see like magic potions have made it all across, or people make a bunch of art of them, they have Japanese magic potions named after them. So if I can see my morphs start to make it across and into other countries, it would be pretty cool.
That would be. I have to apologize that my camera's dead. My microphone's still working, but for some reason my camera died. So you're in solo layout.
It's all good. I don't know if anybody's popped up any questions or things, topics they have.
I've sort of been ignoring the questions because we were so busy talking about things. But if you do have questions, everyone, especially questions that I missed and you want to catch those in the next few minutes, then go ahead and throw those up there and we'll try our best to answer those. Definitely. And I'll scroll through and see if there's anything that we can hit right now.
Do you have any crosses or projects that you're wanting to try and do now or in the works?
Well, I've thought about, since I do have the how nows, and I do have the orange lavis, I've thought about trying to replicate the same effect as the Colby jacks, but I kind of want to just get Colby jacks because I feel like they've been refined for a while, and so there's some higher expression stuff out there, and I'd rather get that than worry about trying to do that. But I'm also interested in playing with some other things. I have the Lucy's that I got from Kyle, Porcello Scaber Lucy.
Oh, yeah.
And he says some really interesting things happen genetically with those. So maybe crossing those with something. I have some yellow lines of Scaber that I could play with the Lucy's and see what that does.
He has shown me a picture. He made a yellow calico pied somehow.
Whoa.
So it was from the orange calico, and it was really nice, yellow and black dots, and then the middle was missing. He's also made some, which I'm trying to make into, he calls it the rainbow, or he used to call it I think it was quad color gene, where it's a Scaber that's got kind of like an orange dots to it, a yellow calico, so it's kind of a yellowish body. So it's got white, black, orange, and yellow in it, so that's why I gave it the quad color name. And then I've tried to mix that with pied and trying to get the babies out of those. He also has one called Sherbert, which is the males are more darker orange, and then the females have this light orange speckling to them. So it's kind of like a calico, but with a white background and orange spots onto it. I've only just gotten a big mixture of them. I didn't do the right thing of pulling the double recessive babies and then inbreeding those. So it's just kind of churning, and hopefully I get a visual pop out of those. That's similar to what I did with the Lava Dalmatians, but they're easy to find. So I've been able to pull out the visuals from those. I'm going to have to start to isolate the Niscus ocellus double het soon, and the Vulgari yellows that I have. So those will be some new things that I'll try to isolate and hopefully get some pictures shown.
Awesome. Yeah, I'm looking forward to seeing all that and the progress of all these projects.
If you can, the most fun or the most visually pleasing morph is that Maples Mardi Gras, the Niscus ocellus. That would be a fun one to reproduce. I had talking about it for a while with others, and I know some other people have made that same cross. I think I was just one of the first to show the cross results. But then in the middle of the project, when I had the double heads, Jeremiah was like, "Oh, cool! Look, Dalmatian maples." I was like, "Oh no, it's hard now, but we can have a couple lines."
Yeah, it's probably a good thing to have a couple lines in the hobby there. Yep. So Kawaii Yokai is asking if we can see the Isopod plushie and ask you where it's from.
This came from a girlfriend, but I think it's the only one of them that's out there. You can probably just get it on Amazon, I think is where they found it.
That looks like a Bathynomus, isn't it?
Yeah, kind of more deep water isopods.
Yeah, the deep sea species. That's awesome.
But,
so that was on Amazon, you said?
Yeah.
Cool. So Heaven was wondering about inbreeding with isopods. As you're isolating these morphs, how much do you worry about that?
There have been people that have posted about this, just worrying about it. With insects, they don't have as much genetic drift, I think, with them. A lot of species, they don't suffer from negative consequences from inbreeding. So you can have just maybe one male, one female, cross them, and then those will just create a line down the line. They don't really show defects, I would think. The only defects I've seen is sometimes, like a Vulgari, their skirts will start to curl up. And I don't know if that's a result of inbreeding, or some people say too high of humidity will start to get those kind of curling. But I haven't had any issue. Some people may say that their colony crashes could be from inbreeding and not having enough diverse genetics. So one thing that I did worrying about that, or trying to prove it out as well, is I took some of my orange pied and I mixed them with 3 different orange females, one that came from a wild type and lava cross, so it kind of had some darker brown markings. So I have that line makes a kind of a dark orange pied, where it's kind of got a little bit of brown or black striping to it. I took one of the lottery or the Spanish orange females, and I mixed that one with the pied, and then I've gotten that line out, and then the U.S. orange with it was some other kind of an orange one. I don't know. I mixed them all. 2 of them worked out. One of them just got a muddled mess, and then it really proved out, and that crashed. But that's how I've tried to reinforce that to help get it a little bit stronger. And I had reintroduced some of those pieds into the this mix, and that's why I think I got the orange pied Dalmatian gene to pop up. So the Dalmatian gene went in there. And I just kind of pulled visuals when they're young and sent them out to people, and they have much bigger colonies and more proven out, because I've dipped into that colony a couple times. And now I just want to try and rebound. But as long as it's out there and exists, there's backups. So as you were saying, the Colby Jacks, somebody that's made them really well. Kyle's working on refining it. He might be a while before he gets them sent out. But Julia, I sent it to her. A friend who kind of keeps a backup for a lot of genes there.
Cool.
You could ask her if she's ready to send them.
Yeah, I would love to get a hold of some of those. They have a really unique look to them. Just I have never seen anything quite like that with the glow, like you're explaining, like that interior glow. Very cool.
So, Isovic, I think. Very curious about the cherry Sakura blossoms that you mentioned a couple minutes ago. Where they originate from, seem to appear in Facebook groups, but never any info. So I've gotten a little bit more info inquiring with the people that made them. They're being posted a lot by Art Garden. They came from papayas, and somebody located in China, I think Taiwan, has developed that line, or somehow they popped up, and it's just a real high pink expression. Maybe it's just a higher pattern to papayas. I have not seen anybody else outside of China kind of post them, so I know they were trying to sell them. I don't know who has them anywhere else. I'm wondering if they might be like it's dyed or a certain type of food that they're fed. I saw in a picture where it kind of had these little, it was all pink except for these dots of lighter pink or white on them. So I'm like, did they just dump it in dye and now showing us what's left over? Any like of the real babies, but sometimes there's like a solo picture that may have been the first one, but somebody had been breeding them for a while and getting a large number of them. So then they can send them out before announcing that they were made. So that's why it kind of seems fishy if they're real or not. I'm just waiting to see them, like somebody get them in person and breed them one generation to make sure that they prove out and it's not just some trick. So we don't really know yet, but I know that they come from China. I think Taiwan was the location.
Cool. Yeah, I hope those end up getting here and they're actually a valid morph and not just...
It'll be popular.
Yeah, definitely.
In Australia, we hardly have any morphs or anything. It's a fresh slate. Can you offer any advice for a community that's essentially starting from the beginning? So if you do find any wild types, maybe reduce down to just like a single gravid female. The brown ones instead of like the dark gray ones, probably a better chance, and you might get a random recessive morph to pop out of them, like leucistics will pop up and show up. A piebald, Dalmatian, they'll just pop up out of the random stock. Or as we found here in the wild, we find a lot of like Mardi Gras looking Aniscus ocellus, and I think somebody in Western Europe has found a Dalmatian Aniscus ocellus, so it's not Mardi Gras, it's just a really fine dot pattern. So I'm waiting for that to make it over here, cross it with maples, or they're wanting to cross it with an orange maple to get that orange Dalmatian Aniscus ocellus. So that would be interesting, but it just, if you do find that wild, shiny one in the wild, you need to pick it up, isolate it, or breed it with another wild type if you just find one, and then find those recessives, inbreed those, get more of that visual trait, and then you can start to create some different versions of them. So find your orange morphs, find your piebalds, your Dalmatian. If it's got a koi patterning, those are going to be the main ways, and just get lucky if you're going to taking wild stock, and you can start to line breed them, just trying to get more brown versions or more high yellow versions if they got the patternings. And then those brown ones might turn into oranges. And as they start to inbreed and they all start to have that kind of inbreeding genetics, a smaller and smaller pool, you might get some of those genetic mutations to pop up that would then show up to have that leucistic or piebald version that pops up. That's probably how we're finding some of these in the hobby, is just they've been in the hobby so long and starting to have the genetic mutations that will interrupt pigment production.
Right. And to a large extent, it's a numbers game. The more you see and the bigger the population you're working with, the more often you're going to get there, or the more likely you have of encountering something.
Yep. Just have to be careful. Lots of enhanced photos, Photoshop, and scammers in the pod community. Yeah, that's where the Sakura ones were kind of worried, because Art Garden has done some really heavily Photoshopped and boosted colors before. So I don't know if the cherry or Sakura blossom, those ones are really real or not. So we'll have to wait and see. But if you've seen them in multiple places and some more realistic lighting photos and such, especially if you have the whole colony shot where you see little baby ones that have it along with the adults, just in the different lifestyle stages, that would be a better. If you can get video of it, though, that's also a good. If you want to ask for video of it taken,
That's
that
a lot harder to fake.
and keep you from scammers that are like, I stole a photo, I'm going to post it. And you're like, can you show me a video with today's date in it? So like show for real that this was a newly captured video of what they are.
Right.
Which Isopod do you want that you don't have? I want to get the orange Hoffs and the whiteout Hoffman's egg eye so I can make that yellow morph. Probably also some spiky ones. I tried the Cuban spikies, but they didn't make it well. They have ones that they look like Ninja Turtles or Shredder. I think they have them on isopods.com, but all those kind of Central America spiky ones are the ones that I would more want to see. They just interest me, but I think they're going to be so hard to keep.
Yeah.
A lot of the different ones. So this one, they don't really have the same coloration, but those would be pretty nice to have. The keys that we're not really supposed to have, but they've made it to the States somehow, and they kind of circulate around, let's say.
Yeah. It seems like there's a lot like that in the hobby these days.
And then I don't know. I want things that don't exist, like the Ukraine or the
orange Ukraine pied.
Yes, that would be a good one too.
Yeah, I'm hoping for that for sure. That would be amazing. I really, like you said, the pattern on the Ukraine pied is so unique. Nothing like that anywhere.
Very, very high contrast with those and high variability.
Exactly.
This is the one that I think I want to get the most. This is the one that looked like, it was kind of like a Shredder.
Oh, yeah.
Turtles is what I'd said it called like. But I think they have those, or the isopods.com has some. I'm just trying to get them to reproduce now.
Wow.
I wish
gorgeous.
Indeed. I wish I could keep the Marinella species. There's a lot of different morphs that are coming out of that, like more and more pop up, but most of them are overseas.
Yeah.
We have the lava morph and other things. Here's one of Kyle's lava pied that he's made.
Oh, very cool.
Much better expression than the ones I have, but it's still following the same rules of pigment on the head, pigment on the tails. So I think those, I haven't seen any more results from his that are out there, but here's an example of an older orange pied where it starts to get that more white expression with just a little bit of flaky orange coming through
on the edges. Yeah.
As they orange, they start to get like that. I've had it where it's just completely looking white over it, but it has that kind of glow coming underneath, kind of how the Colby Jacks are.
Okay.
But this was another morph that I was working on. I lost a lot of them, but I'm trying to backbreed them again with things. They are what I called gold dust pied. So it's a yellow calico mixed with an orange and pied. So the babies came out, they'd be orange or they'd be calico or they'd be pied. So it's kind of like an orange pied, but it has these dots in it as well.
Oh, I love that.
A yellowish goldish trait to it.
That's beautiful.
So I tried very hard to get that one to prove out. I had like 6 visual females, but they never, I think they crashed on me and stuff. And this is one of Kyle's lava Dalmatians. These were some of the photos that inspired me to start to make my own line of them.
Oh, wow. Yeah, you can see it in the larger spots.
Yep. So those are nice. They're what kind of inspired me to make some things. Here were the original 2 orange pied that I pulled out. So I just like how very stark they are whenever they're younger.
Yeah, I think those are the photos I think you sent me originally, like a long time ago.
Yep.
You said, I've got these, and I just said, those are amazing. Yeah. I love the clean contrast on them.
Very, very much. That's what inspired me to start to do that more of genetic stuff that I can make my own crosses and things. I think these were, I showed it earlier, some of the first yellow pied that had popped out that I was starting to isolate. So this one's just without pied. This is that yellow gene and with yellow pied. And that's where I've started to make that bin too, of any of them that showed up.
And do you notice that most of the pied individuals, well, I think you mentioned something about this. A lot of times the pied individuals won't show the full pied trait until they get older. Like they look solid colored and then they slowly lose the pigmentation in the center.
They can. Some of them, when they're really young, they'll have that straight pied. And then I think some of them will develop it slowly over time. So they get more of that pied. That's why I think there's a lot of small, regular looking ones in there. I'm just leaving them alone for now. If they grow up and they get more of that pied trait. This is one, it's probably hard to say the colors, but this is one of that quad color that he was talking about. So it has some kind of white and black traits, some yellowing, some orange dots on it. It's probably not as good of a photo, but I'm trying to add that into pied.
Yeah, that's a very interesting effect in there.
I think he's also calling them rainbow. Here would be, I think those two original ones that I showed a minute ago, as they aged, they start to develop more of that orange would start to come through a little bit.
Encroaching a little bit, yeah.
So this is like the older version of orange pied. When it gets a little bit older, they'll start to gain that trait. And here is the lemon duckies that I had got.
Oh, wow.
So they are really, they don't have any of that dark middle band, but they got kind of a whiter skirt and just yellow jelly beans.
That's a cool effect.
They're pretty nice. And what I like is some of the lava pied will get this really high expression Dalmatians out of them. So that's what I'm trying to concentrate more on, the lava Dalmatians. I'm trying to find the, so here's maybe some more Colby Jacks as they were isolated.
Yeah, loving those.
And my friend's orange pied colony. So they'll prove out, or they'll start to prove true there. And they get really nice. I'm trying to see if I've got, these were some of the first lava Dalmatians that I had that I isolated. So
you can really see the three different colors on there.
Here are the pink duckies with the lemon duckies. These were their next generation of babies. So as you can tell, they're the white ones, and then they have this kind of a pinkish hue.
Yeah.
Brown. So we'll see if they're sexually dimorphic. I just put them both in there because I wasn't sure and let them grow up together.
Mm-hmm.
So hopefully we'll get some pink lemonades and some lemons out of those.
Yeah. Yeah.
This was one of the first visual. Had all these double hets and maples. So the first maple Dalmatian in there. I was able to be able to pop him out. That was pretty neat.
Yeah, the color on that, that's beautiful.
My favorite project that I had come across, let's say. But
so are these photos that you're showing us? Heaven was asking if are you using a macro lens or just your phone?
Just my phone, just kind of zooming it in and trying to take a shot. I would appreciate a macro lens, but I think I've tried a couple in the clip-on ones. Here's one.
Oh, go ahead.
One of the yellow calicos that I found in the wild in the U.S.
Oh, wow.
But I've never gotten that same kind of high expression. It must be just due to the wild traits that they're in and different locales, because most of the babies just came out very low spotted.
Mm-hmm.
And it
still
kind of
cool, but that original one is amazing.
Yep. Along with this one, I found a red skirted. I think maybe now they're probably called Rusty or something, but Kyle did have a line that he called Dragons, Red Dragons. So I used that name, but it died out, unfortunately. So sometimes you can get some oddballs to show up, but they don't really prove out as much. Just some weird patternings, different things.
I like that though. Yeah.
Come through the line. This is one that didn't prove out, but I like how it's looking. It's kind of a mixture of the orange and normals or screwball tries they have. So some different fun things that I've been able to create. They're don't know. There's some fun looking. Another fun pied one was
Look at that. Oh, that's amazing.
Prove out more, but
Yeah.
you just got to roll the dice with those pied looking ones. Oh, here is a very, very old orange pied, where it just has pigment left on its head and everything else is kind of like under the translucent layer.
Almost like the Colby Jacks.
Mm-hmm.
Yeah.
With age, they'll start to lose more of their orangish and start to develop that. It might be out of the fun photos that I've got. I'm trying to look for the gold pied that I had. Another line that I put my hands in is the Valencia, Porcelia Valencia. They would come in these, every so often they'd pop out a yellow one or an orange one. So I isolated just the orange trait, and they look like mini Magnificus.
They do. They really do. That's cool.
At the Seville line. And whenever they were younger, they look kind of like Nicolesi, I guess.
Yeah. Yeah, I can see that.
Been compared to. But hopefully they're still out there in the mix. I've given up on that project to try and just move on to other things, since that one was just a line breeding or isolating of them instead.
Yeah. Well, you've got some amazing things going on. I am so glad you came to join us. There's so much out there that, and so many of these are what many people would refer to as, just the normal isopods, or that they're ones that are easy to ship. You don't have to worry about crossing state lines with them. You can get permits for them. Mm-hmm. And that's what I'm trying to focus on, is the species that you can easily ship across state lines. And they're so variable, and there's so many different possibilities. I love it.
Yep. So one that we might see out there, that's another of the lottery ghosts. They've got that purplish, which is, I think, just a het orange. And I think that purple ghosts, or the Porcelio ghosts out there, are just a het whiteout. So they kind of look like that. But that's my taking on them. I've seen sometimes they pop up like little bitty pied purple parts to them. Mm-hmm. Ghost ones. So I don't think that's a really proven out line. Here is an adult one of those pink ones.
Oh, wow.
And ducky. Kind of hard to get the right coloring, but they look kind of a little more orangish, a little bit brownish as they grow older.
But still, that's amazing.
It's fun to work with, and if you find any that you like out there, just try and find the morphs and cross them. I think we have more morphs of these local U.S. species because they've been in the hobby longer. More have popped up, and we've been able to find them. And if there's something out there that you want to cross, just try it, work with it, and it puts a lot more fun into them instead of just keeping them. Yeah. They'll be the ones that you've made. You've created this new kind of trait, and it's your own line. And if it's something nobody else has, you can name it.
Yeah.
Kind of give it another street name than the morph name. But a lot of them out there, which I just want to use a platform here. If you have a whiteout morph, please give the species name, and then you call it like T-negative albino glaciers. Because if we just start calling them Cubaris glaciers, we're going to have the red edge whiteouts and the marina whiteouts and
the king whiteouts.
It's going to be really hard to delineate them.
Right.
It's already started with magic potions. The Armadillidium granulatum had a magic potion morph. And I saw somebody post up, is this the Vulgari or is this the granulatum magic potion? Which I propose a name, just call them grand potions or granulatum.
Oh, I like that. Yeah.
It would be good. Just some other name so then we can tell what's the original magic potions and you won't have people confusing them. Because we already have Japanese and US magic potions and you can kind of tell the difference, but if you get them, you're not going to really know for sure if they just call them magic potions. And
exactly.
There's other Dalmatians with yellow on them. I think there's Armadillidium depression. Mm-hmm. Those have a magic potion name to them as well. We can keep the front name with them if you're going to keep the magic potions, and don't just sell them as magic potions if you can.
Right, and that also just points out that the need for maybe when you isolate something, even if it's similar to another line, and has the same species and whatnot, when you isolate it, just like the American line magic potions are genetically distinct population that has slower growth, larger size, things like that, and then has been line bred for the increased yellow coloration. And so when people have the Japanese line magic potions that are smaller, faster breeding, and they're not line bred for the yellow markings, by naming them the same things, you get people who put them together, and then you lose the genetic distinctness of the lines. So that makes the case for if you make a morph and it's the same mutation as far as you can tell, but it's a different line, that's a justification in my mind to give it a different name.
Yep. That's where Kyle was trying to think of what to do with the yellow ornatus, the witch's brew. So I was thinking, like, what's another name for a magic potion? So we got witch's brew. I want to have Midas's elixir, wizard stew, just kind of some names that would be fun if that morph pops up into other things.
Right. And the Porcellio expansus that has a similar trait, they call it the witch's potion. so that's a different, distinct one.
Oren came out with those, and it's on par. It's great that he gave it a similar but different name to him.
Distinct. Yeah, that we need to make. That's why I call my orange pied P. pruinosis. I call them freckled gingers, which Richard from Tarantula Collective suggested the name. I love it because mine probably have some, because they came from different stock, probably have some distinct traits. And there's an orange koi version of those now, which is separate from the orange cream.
Koi is the UK version, or
right? The red koi. That's also in the Philippines. They have it there too, and it's red koi, which is a little different. So it's important to keep these names separate because they come from different stock, may have different traits. Whether or not we recognize that at first, keeping those lines distinct is important. So
I think I've got my orange creams. I've got kind of like what that pied you're talking about, or maybe it's just more high orange expression with just a little bit of white on places. So that's what I'm line breeding for. I'm just trying to get the highest orange concentration instead of some of them I've seen have got very few orange dots to them. The orange creams, and it all depends. Those are sometimes like they can kind of line breed, but they'll be very variable unless you kind of cull them out. But if somebody's selling them, they're going to probably sell the less visual ones to you, so you get a less hopefully they can be nice and give you some nice visuals as well, instead of just trying to cull out the ones that they don't want to keep. So fortunately, that's sometimes how it happens. And I think the Scaber orange Dalmatians that are out there, they've just gotten so diluted that sometimes they'll come up just all white, or I've seen even maybe a T negative pop out of them. I don't know if I had a tainted line of them or something, but they just started getting less and less orange dots on them until they started popping out with no eye color from Inbreeding. So,
yeah, I've noticed that too with the Dalmatian lines. They tend to lose expression over time. Some of the first photos of the first Briceville Dalmatians you see are amazing.
I got some of the Briceville stock, and the ones that I had pop out looked like leucistic. They had no other dots on them except their eye colors. But I didn't really work to try and isolate and inbreed just those. I thought it was just a fun mix. So it had just the wild types mixed in with a couple of those. But I think I've seen where you're talking about where just that real high expression of large black dots, and then it's the small black dots that is with that US Dalmatian line, where the orange Dalmatian line that showed up, not the one that you made where it was the cross between the orange and the Dalmatian. Those had the little tiny orange dots, which I think are much better representation of the Dalmatian gene.
Yeah, and that's I guess one thing that at the time when I isolated them, I didn't think I should call this line something different. I just called them orange Dalmatians. I should have called them something distinct. And I could have had orange Dalmatian in the title, but I should have put something else in there, like a Quermax line or whatever, something to distinguish them. And at the time, I didn't think about it. And so there are probably people who are mixing them and have mixed them and whatnot. And it's kind of sad. Had I known at the time what effect that would have had, I would have done that differently. But
yeah, my dream line that I'm going to create one day is like how Oren's got a calico. My cat is named Oran, kind of like a short form of orange. So G. So I want to have Oran's calico or Oran's pied. Them out there, and they'll not really get them confused, but it'll just kind of be
an homage, kind of.
Homage to that line that kind of helped start it all. Because I think Oren McGonigal was one of the first pioneers into the hobby.
Oh, definitely. Yeah. Father of the isopod hobby in a lot of ways, for sure.
The only textbooks that are out there were for a long time.
Yeah, very true. And I'm just going to put a plug out there for the Tunisian pied Porcelius caber. We need to get those out here.
Yes,
those are great. They are fantastic. I hope they still exist.
Same. I have some saved photos, but I haven't seen anybody else post them in a while, so they'll be nice to see.
Yeah. Well, it's coming on 7:00. I need to kind of close up here, but I want to thank you again for coming. Really appreciate it. It's been a lot of fun. A lot of people learned a lot of things, including me.
Cool.
Do you have anything else you want to say before we close up?
Just have fun with the hobby. If you are no longer interested in it, try downsizing. And if you go too quickly, you'll start expanding. Like I had 150 different bins, maybe 100 different species, and that just causes burnout. Mm-hmm. Try and pare down to what you love the most and make your own crosses, your own experiments, isolate them from the wild, and you'll have more fun with it. And it'll start to carry along that hobby. So it can just be a slow burn project, because these can take, 6 months from the baby to get to adulthood to then have babies, and then those get up the double recessives and those have babies. So it could be a year or so before you even get any of those visuals that you're looking for, and then another year before you have enough of them to start to distribute. So it's a long-term project, but if you just focus on the ones that you want to keep and maybe keep the numbers low, I may have now 40 bins or so. So it's a lot more manageable, but that'll help keep your interest in the hobby instead of getting that you want every single species. You got to catch them all and then you'll burn out. So don't try and ramp up too much. You'll probably regret it later.
Very good advice. I like that. Very good.
Thanks, everybody.
All right. Thank you. Really, really appreciate it. Thank you. And thank you, everybody, for joining us. And I think this is going to be a popular one to rewatch, too.
Thank you.
All right. Well, thank you. And I'd love to have you on again when you've.
I'm going to get some more proven out for sure.
Difficulties here. Oh, you're back. Okay, cool.
Yeah.
Okay, sorry about that. I don't know what's going on. Some technical difficulty. Thanks again, Derrick. Really appreciate it.